All right, we're almost done with our DNA to proteins. Where we left off was adding amino acids to the polypeptide chain. They are added until a stop codon is reached, so we know not to add any more. That can be somewhere between 100 and 5,000 amino acids. When the stop codon is reached, the ribosome releases the mRNA and it goes one way, polypeptide goes another. Okay, add a little bit more to the notes. There's a whole bunch in the translation section and it looks kind of small, so you might have to zoom in. This is a great summary of protein synthesis. So we've got our double-stranded DNA molecule, the entire owner's manual. We're going to pick out one gene, which is one specific thing we want to do, one function. And we open it up, we have the DNA strand. We're going to transcribe it, make a copy, and mRNA, and then that is going to be translated into a protein. Okay, some ribosomes attach to the endoplasmic reticulum, which is another organelle of the cell. When it does that, that is just to help the protein in the folding process. The folding process gets pretty darn involved. The protein sometimes needs to be modified, and the folding happens in, there's 4 different degrees to describe protein structure. That's maybe a little bit more than we need to go into right now, but the protein, the final shape of the protein is what makes it functional, and it's very important. So where are we? Let's see if we've got this. What kind of nucleic acid is made during transcription? That's the RNA. How is mRNA different from tRNA? All right, mRNA carries the genetic message from the nucleus to the ribosome where it's translated. tRNA carries the amino acid to the ribosome, and it has an anticodon that binds to a specific codon on an mRNA. Which amino acid is coded for by the RNA sequence CUA? So I find the C on the left-hand side. I'm going to go all the way over to the A. That is, okay, A, and then I'm going to look, sorry, CU and then A. Oh, right here. All right, leucine, right there. What does the start codon AUG do? 2 things. Starts the translation, and it also can code for methionine. Briefly describe the 3 steps of protein synthesis. 1. Genes in the DNA are transcribed, copied into RNA. That happens in the nucleus. The mRNA is translated into a polypeptide chain with the help of ribosomes and tRNA. That happens at the ribosomes. And then last, the polypeptides combine and are folded up to form proteins. And that happens in the ER and the Golgi apparatus. Okay, last bit is about mutations. We want to know what causes mutations and describe the type of mutations that can affect genes. All right, so what is a mutation? It's any change in the base pair sequence of DNA, and it can involve a large part of the chromosome or just a single base. So I'm starting a new little section on my notes. What causes mutations? Before a cell divides, it has to make a copy of its DNA so that each new cell has the same DNA as the parent cell. It's got the entire operating manual. Sometimes there are copying errors during base pairing of DNA, which causes mutations in genes. Some of these occur by random chance. Others occur as a result of physical or chemical agents. These are called mutagens. In plants and animals, random spontaneous mutations are rare, and there's proofreading in place there are proofreading mechanisms that help lessen the chance of having spontaneous mutations. But the mutations that are caused by mutagens, some of them can lead to cancer. Those are called carcinogens. Those are much more worrisome. Non-random mutations. damaged DNA is caused by mutagens in the environment. So what are some of these mutagens in the environment? A lot of them can be high energy radiation, X-rays, ultraviolet rays. There can be chemical mutagens, pesticides, lead, mercury. There's many. There's tons. Too many to list right here. Some chemical mutagens are similar enough to the bases found in nucleotides that become incorporated into the DNA, blocking the bonding of real bases. But most mutations that occur in individuals are not passed on to the offspring. Well, why not? Well, most mutations occur in somatic body cells, not the gametes, and they are called somatic mutations. So the mutations can only be passed on if they occur in the gamete cells, which are what you need for fertilization and to create offspring. Okay, so most of the mutations that happen are going to be in regular body cells. So some human disorders are the result of mutation that occurred a long time ago in the gamete of an ancestor, and the mutation happened, the error happened, and then it was passed on to the offspring, and then they procreated, and it just kept getting passed on and passed on. So some examples are sickle cell disease, cystic fibrosis, Tay-Sachs disease, color blindness, hemophilia, muscular dystrophy. So these are diseases that get passed on right to the offspring. So there's some notes. One of the ones we're going to look at quickly is sickle cell disease. So sickle cell disease, let me just check my next slide real quick. Yeah, okay. So sickle cell disease is a fatal blood disorder that results from an inherited mutation in a gene for hemoglobin that has been passed down through the centuries. The original mutation was just a single base. That was the only one that had an error. And what happens is the mutation results in a deformed hemoglobin molecule. And then that means that oxygen can't properly bind to the red blood cells. So this is just a little bit of notes. Back to sickle cell. So normal red blood cells are round like donuts. So on the screen, the ones that are round like donuts, those are your normal red blood cells. They bind oxygen, move it through the body. The sickle cell blood cells become hard, sticky shaped. They look like sickles or something that's used to cut wheat. And when they form this way, they're hard and pointed, and they can't travel through the small blood tube, and they clog the flow and they break apart, and this causes pain and damage and low blood count and anemia and lots of problems in the body. So mutations within a gene fall into two general categories. You can have base substitutions. This is when a single base is replaced by a different one. These are usually not as bad. They're survivable. They can be silent. You may not know that there is a mutation, or they can be beneficial. But the main point here is it affects only one base, so it really only affects one amino acid. So if you look in our diagram, we have our DNA sequence, mRNA, our polypeptide. If we have one substitution, and they have it highlighted here in green, at the top I think it's a C, and down here it gets changed to a T. That switches out one amino acid, but from the rest behind it is all going to be the same. So it's a small change. All right. Now, the more worrisome mutation is when you have insertions or deletions. These are more disastrous, and they're almost always harmful because it affects everything that happens afterwards. So on the other side, the B example, we have an A is taken out. That shifts everything over. So now, instead of having one amino acid that's changed, everything afterwards is changed. So sometimes this means that the protein is not functional, we can't even use it, or it's completely different. Both of those would be a huge problem. So here's a little bit of notes on that. Let's just go over this briefly, and then we're all done with this. Explain why a base substitution is often less harmful than a base deletion or insertion. Okay, if we have a base substitution, you are only going to affect one amino acid on the polypeptide. But a deletion or insertion switches the whole view that we're looking at. So it affects all the amino acids downstream from where it occurs. So it makes that protein either non-functional or completely changes the function. Describe how a mutation can be helpful rather than harmful. A mutation could be helpful if it alters the protein in a way that benefits the organism and its environment. So you could have a mutation that provides an animal with different pigmentation that has better camouflage, and this mutation would probably continue on because if you have better camouflage, you're less likely to be prey. Your offspring will have a higher chance of survival, so that could be a helpful mutation. Give an example of mutagen. We've got UV light, X-rays, nuclear fallout, chemicals, all those kinds of things are mutagens. And that's it. Done.